Hello, my name is Sebastian Truong, and today I will be presenting my work on the effect of Pseudomonas stutzeri on the inhibition of Alexandrium tamutum, exploring Rhamnolipid biosurfactant amplification. To start off, 10 to 20% of coastal waters globally are affected by harmful algal blooms, or HABs. According to Mission et al., these harmful algal blooms start from commercial fertilizers that deposit nitrogen into groundwater systems such as the Gulf of Mexico. Blooming algae begins to decompose, in which aerobic bacteria aids in the breakdown of the organic compounds. This creates hypoxic conditions, which is a lack of oxygen in the water, and results in the creation of dead zones, which is areas with so little oxygen marine life can no longer be supported. Boyd et al. Explains that blooming Alexandrium cantonella produces a saxitoxin. Saxitoxins cause paralytic shellfish poisoning, or PSP. PSP disrupts the shellfish life function, contaminating food supply and causing environmental and economic damage. Su et al. Explains that Pseudomonas sp48 is able to inhibit the growth of Alexandrium tomarense, another HAB-related species. It breaks down the cell structure in Alexandrium tomarense by excreting algacidal compounds. However, Pseudomonas sp48 only inhabits the Donghai Sea in eastern China, which led me to my next study, Cowan et al., which explains that Pseudomonas stutzeri inhabits soil, freshwater, and marine environments, which means it is able to be applied to greater field value. Pseudomonas stutzeri is found in Indian lignite coal beds as well and releases rhamnolipids in this environment. Rhamnolipids are biosurfactants that are able to clear biofilms off surfaces like rocks in aquatic environments. This led me to my research question: to what extent does Pseudomonas stutzeri inhibit the growth of Alexandrium tamutum, and does lignite coal supplementation increase biosurfactant rhamnolipid yield? This is because Alexandrium tamutum is a non-toxic rather than toxic species of algae. My first hypothesis is that P. stutzeri will be able to inhibit the growth of Alexandrium tamutum before it is able to create a significant bloom. My second is that P. stutzeri will be able to produce greater amounts of rhamnolipids when exposed to greater concentrations of the lignite coal. My experimental design is simple. My prerequisite standard curves were made, which funnels into two assays. Assay 1 is a chlorophyll concentration before and after test, and assay 2 is an L-rhamnose quantification and organelle assay. These two assays funnel into data collection, which is used to assess and draw conclusions. I started with my prerequisite standard curves for assay 1 and assay 2. Assay 1 details a absorbance at 665 nanometers versus chlorophyll concentration in milligrams per liter. Assay 2 details absorbance at 421 nanometers versus absolute L-rhamnose concentration in micrograms per milliliter. Assay 1 starts with chlorophyll concentration before and after test, in which I took the baseline absorbance of a sample of a tamutum in a cuvette and ran it at 665 nanometers in a spec. Then I administered treatments to each of these cuvettes in 35 PSU salt water, which is a control, a 5% solution of commercial algicide, which is my positive control, P. stutzeri in its marine broth, which will be the experimental group, and I administered all these treatments at 125 microliters, 250, and 500 microliters. Then I separated the supernade from each of the treatment groups by running through the centrifuge and using micropipette, and I injected into another cuvette to run the post-treatment absorbance at 665 nanometers again, subtracting the values and then interpolating with the earlier standard curve to acquire delta values. My second assay is an L-rhamnose quantification and organelle assay. I first created groups using a basal medium, which is a medium sterile of carbon, and adding alternative carbon source in the form of a naturally occurring lignite coal. I started with 0, 1, 2.5, and 5% lignite coal groups and cultured P. stutzeri. After the culturing, I removed the supernade after centrifuging and added it to a glass cuvette. I then added an organelle reagent made from H2SO4 and organelle powder. The role of this was to break down the rhamnolipids into L-rhamnose, which is a byproduct. Then I ran the samples through a spectrophotometer at 421 nanometers and put it through a boiling bath, which would turn the clear solution into a cloudy solution, which I can use to quantify absorbance. I then interpolated with the standard curve to make these values what you see here for assay 1 and assay 2. After acquiring the values for assay 1 and assay 2, I ran a one-way ANOVA, which returned p-values in magnitudes of negative 10 to the negative 5th and 10 to the negative 7th, which indicates potential for statistical significance. I ran two-tailed independent t-tests for both assay 1 and assay 2. Most notably, in assay 1, 5% algae off and P. stutzeri showed no significant difference with a p-value of 0.869, which indicates that P. stutzeri performs at nearly the same level as a 5% solution of commercial algicide. And for assay 2, the 2.5% lignite coal group and the 5% lignite coal group showed a significant difference with a p-value of 6.525 times 10 to the negative fifth, which indicates a significant difference, showing me that as you increase lignite concentration, the amount of biosurfactant produced will increase. My bar graphs show very similar trends in both P. stutzeri in assay 1 and the lignite coal concentration versus L-rhamnose concentration in assay 2 with a bar graph and scatter plot. After drawing the conclusions, we confirm hypothesis 1. P. stutzeri will inhibit Alexandrium tamutum just enough to prevent a significant bloom. And hypothesis 2, in which P. stutzeri is also able to produce more rhamnolipids when it is exposed to lignite coal in the medium. However, you do have limitations here in which methodical limitations might apply to the unintentional lysing of chloroplasts during the centrifugation or the cloudy byproduct of algae breakdown, which can mess with my spec readings. Here, in environmental limitations, we have the emergence of a mutant algae strain and possible disruption of nutrient cycles, which are both environmental risks associated with introducing the compounds to an environment. Then I have future implications, which opens the door to a new class of sustainable biocontrol agents. Pseudomonas stutzeri culture with lignite coal could become a vital tool in protecting marine ecosystems from HABs, in which creating an algicide from the natural occurring rhamnolipids might become a solution to replace commercial chemicals, and lignite coal will increase the rhamnolipid yield in production of such algicides. Using P. stutzeri to mitigate small population in smaller ecosystems will allow it to gauge the environmental impact before we release it to oceans. These are my references, and thank you for your time.